Good morning, everyone. Welcome to our webinar today. Our webinar titled Protein Sample Prep, Be Gentle, Stay Cool. Before we begin our webinar, we have some housekeeping to tend to. So I'll just shortly present the housekeeping, then we can move on. So the webinar will be recorded today, and at a future date, you'll be sent the link to access the recording. Also, you are currently and will be throughout the webinar, your video will be off and your audio will be muted. So if you do have any questions, please type them in the question pane on the right of your screen. And we have some moderators here today who will be answering those questions as we go live. And also at the end of the webinar, we'll have time to have a Q&A session, again, using your questions in the Q&A section. Lastly, today at the conclusion of the webinar, there will be a post-event survey that you'll be able to fill out. Please, your feedback is always welcome. It helps us make our webinars bigger, better and greater for the future for you. So today you'll be hearing from our speakers, and our number one speaker is Peter Tran. He is our field application specialist in the proteomics space here at Merck. He is based out of Sydney, and at the moment he's not doing any customer visits, but he's doing lots of virtual visits and virtual communication. So if you do have any questions for Peter at a later date, please feel free to contact him. You'll get his contact details at the end of this webinar. Also, your moderators are Dr. Yi Chen Ko. He is our marketing specialist, again, the marketing specialist in this space of proteomics. And you're currently hearing from me. I'm your field application specialist manager here at Merck. So again, we're here today for protein sample prep webinar. There have been 3 previous webinars to date, one on winning Western, also from Peter, a qPCR 101 webinar, Peaky Blinders, tips for improving LCMS sensitivity, and protein sample prep today. And in a fortnight, we'll have our last seminar in this series, which will be 3D cell culture. If you haven't attended any of the previous ones and you would like, again, access to the recordings, please reach out to us and we'll get you access to that. So enough from me. Without further ado, I will pass on to Dr. Peter Tran, and he'll kick off with his seminar for today. Thank you.
Thank you everyone for taking the time to join us today for the webinar on protein sample prep. Firstly, a little bit about myself. My name is Peter Tran. I'm based in Sydney. I have over 5 years in industry, including 4 years with Merck. I look after proteomics and lab separation. And about my academic background, I did a master of bioengineering in South Korea, PhD at La Trobe University and ANU in Australia, and I spent some months in Max Planck Institute in Berlin. And after that, I did postdoc at the Garvan Institute in Sydney. So far, I have about 15 publications which are focused on proteomics and protein characterization. Today I'm going to walk you through the instructions, stabilization, purification, depletion, and of course they all related to protein. And the last session is the Q&A. Just a little bit about the overview of protein research workflow. We can start with a cell or tissues or a recombinant protein produced in bacteria or mammalian cells, right? You can do cloning PCR to produce the protein, and then we need to extract the protein. We need to make it accessible. We need to lyse the sample and extract protein, and then at the same time we need to protect the proteins from protease. And then some people they want to isolate or purify the target protein further, or concentrate or clean up before they can analyze by Western blotting ELISA, IP, or mass spec. And the first session I'm going to talk today is the protein extraction and protease inhibition. The protein extraction overview. So we start with cells, tissue, blood sample, serum sample, or expressed protein, right? And there are three different steps we need to do for the extraction. We need to homogenize the cell, the sample, and at the same time we need to solubilize the protein using lysis buffer, enhancers, lysozyme, depending on the sample type, whether we need to use the enhancer or lysozyme. And then also we need to stabilize protein using protease inhibitors. And all these three steps need to be processed simultaneously. Then we got the cell lysate, and then we use the cell lysate for purification or cleanup. And the first step in lysis is that you need to determine which reagents and method to choose. And it's largely depend on the sample type, whether it's from plant or from animal tissues or bacteria or yeast. And also, you need to consider the location and abundance of target protein as well. So it's very important that you can choose a proper method to extract the target proteins based on their location. And if you also consider the required protein yield that you need to for the downstream application, whether you need a lot of protein or just a little bit, depending on the sensitivity of the downstream application. And your lysis condition or lysis buffer need to be compatible with downstream analysis like mass spec, Western blotting. Okay, regarding the extraction, there are two typical methods. You can use physical lysis or chemical lysis, or a combination of both methods, right? With the physical lysis, you normally use beating, sonication, French press, or grinding. If you have plant tissue, for example, so you normally do the grinding. Or if you have bacteria, people normally do sonication. And at the same time, if you want to increase the yield, you want to use the chemical lysis in conjunction as well. So you need a lysis buffer, or sometimes you need to do the fractionation just to isolate the organelle. And so this method is some is gentle and some harsh. For example, with the osmotic lysis, it can be gentle or detergents, enzymatic digestion or hand homogenization. They all have a gentle method. And beating, freeze, or sonication range press, they all harsh methods. Okay, so let's touch on a little bit about the lysis buffer. How does it work? So with this question, I want you all to get to the poll that's on the right hand side of your screen. Just click on the poll and pick one of the options that's more relevant to you. What buffer do you use for extraction? Always Reaper buffer, commercial buffer, optimize the buffer by yourself, or you follow publication for the target protein. Just pick one and vote. I give you a few seconds to do this. Okay, that's very interesting. Most of you follow publication for the target protein. That's really good. Good answer. And some optimize buffer by yourself. It might be a little bit tricky, but it would be highly recommended as well. Some using commercial buffer, yeah, it's very easy, right? Just buy and use it because it's already optimized for the sample type. And 14% of you use Reaper buffer. Okay, so let's explain a little bit about the buffer, the lysis buffer, and how you can make it by yourself or optimize by yourself, right? So this is the common composition of the buffer. You need a pH, you need a buffer, you need source, you need detergents, and sometimes you also need the reducing agent like DTT or T-cell. And the composition of Reaper buffer, I show you Reaper buffer because the most common one that people use to for lysis. And Reaper buffer, you have there is the twist, 50 millimolar twist, pH 8. So why do we need the pH 8? So if we're looking at the database for the protein isoelectric point, the PI, PI is the pH at which the protein become neutral or become precipitated or aggregated, right? You don't want to choose a pH that's close to the PI of your target protein. So if you want to look at the switch growth on the left-hand side here, the database, you can see most of protein form two peak at about 5.5 and 8 PI. Or in E. coli bacteria, for example, same thing, right? 5.5 and 8.5, for example. For human, about 6 and 8. But you can see that's very small, or the least protein have the PI at around 7 to 8, right? Between 7 to 8, there, less protein. So that's why people normally choose pH between 7 to 8, we see the physiological pH range for the lysis condition. And also in this pH range, all the protease inhibitor, phosphatase inhibitor, they active at this pH range. And secondly, source, you need a source for osmotic lysis, and you need to have the detergent for solubilization of the cell membrane. And the common one is SDS, which is the ionic detergent, NP40 or Triton 100, which is the non-ionic detergent, and DOC. Okay, so and I want to show you this experiment, how you can screen detergent for the protein solubility. So if you can start with the very basic buffer, with yeast and HCl, and you can add 1% of each detergent, right? in each lysis buffer, you can do the detergent screening and analyze on Western blotting. For example, here, S is the soluble fraction, E the pellet or insoluble fraction. You run both fraction on Western blotting and use antibody to detect the target protein. As you can see, without detergent, with non-ionic detergent, with ionic detergent, there very less amount of target protein can be detected. But with the switch variant or charge, the target protein show a big band here for in this study. So this is a way that you can screen detergent and optimize your lysis buffer. Okay, next one, the enhancer. There's some common enhancer that you can use normally for bacterial sample because they can use to degrade DNA and RNA using benzonase or DNase 1. DNase 1 is a very common one. However, it's only cleave double-stranded DNA only. If you have bacteria, they normally have a circular DNA, right? So in this case, benzonase nuclease. And chicken eggs white lysosome for lysis of gram-positive bacterial membrane. R lysosome, which is for the lysis gram-negative bacteria, normally for E. coli, and they work much better than the chicken egg white lysosome, about 250 times greater than the egg white. And this is the result showing you that using the benzonase nuclease, you can degrade a lot of DNA and RNA up to when you use up to 50 units per mil. And with the treatment of benzonase nuclease, you can reduce a lot of viscosity because the viscosity they are formed by the DNA and RNA material, and you can see the viscosity reduction significantly using the nuclease. And this is an example of you see a comparison between the home brew buffer HP and the commercial buffer. And when we add the R lysosome and benzonase, you can see that you can extract a lot of protein. This is the total protein concentrations. You can extract much more protein, about 3 times, when you use the R lysosome and benzonase. And when you run it on the SDS page, you can see this very significant signal come up. Okay, let's move on to protease and phosphatase. Protease and phosphatase play a key role in cell signaling. They strictly control the cell signaling, and especially for the phosphatase, when the phosphatase can cut off the phosphate group from the protein and make it dephosphorylated. Or the other way, the kinase can produce or introduce the phosphate group into the protein. So this is how the enzyme can transfer the signaling between the protein or between the organelle within the cell or tissues. However, when we lyse the cell, right, we at the same time we also release this protease and phosphatase to the environment. Especially when the lysosome, we see an organelle within the cells, this contains a lot of nuclease, phosphatase, protease, and your lysis buffer might lyse lysosome as well and release this enzyme. And then this enzyme will degrade the protein. So that's why it's very important that you have the protease and phosphatase inhibitor in your lysis buffer at a very earlier stage, just to protect your protein. This is an example of a product for the protease and phosphatase inhibitor. It's called mass spec safe protein and phosphatase inhibitor cocktail. And normally you see there's a combination of different inhibitor in the product as shown here. And if you can realize that there's some product they there is EDTA and there's some EDTA free, right? I will touch on this one in the next slide. Okay, so what are the common proteins in the cell or tissues? So basically, there are serines, cysteines, metallose protease, which are the most common one, and also there's some aspartic and threonine protease as well, which are forming the family of protease. So we need to have the inhibitor that can inhibit all of these one. And the most common one is the PMSF, right? They can inhibit serines and some cysteines in protease. So some inhibitor can inhibit specific protein. That's why you need a combination of them. And with the EDTA chelates the metal, magnesium, or iron genes. Therefore, it can inhibit metal-dependent protease. However, if you do the lysis in conjunction with nuclease, right? I would not recommend to use EDTA because it also inhibits nuclease. Instead, you can use EGTA only chelate calcium. And what about the phosphate inhibitor? So the most common one are threonine and serines. Phosphate that's present at about 10% and 90% respectively in human sample. And the most common one is the sodium octanovanadase that can inhibit tyrosine phosphate, or sodium pyrophosphate can inhibit serine and threonine phosphatase. When you buy the product, just make sure that you check the composition of the inhibitor, and then you will see how it works, how it inhibits the phosphate. Okay, just a quick summary about the first part of my talk. I have touched on the lysis buffer, pH, detergent, how you can screen detergent, enhancers, benzonates, lysozyme that you can use to increase the yield of extraction, and stabilization using protease inhibitor, phosphate inhibitor. And there's some tips that you can take from this the first section. Just to make sure we buffer, we need to pH that's suitable for your target protein and also for the inhibitor that you use. Be cool, just always keep it cold because the protein doesn't like heating. Be clean, just always use the fresh sample, fresh buffer, and clean sample. And be fast, you need to process as fast as possible because the protein will degrade very quickly. And be gentle, use a gentle methods, and they need to be compatible with downstream analysis. Okay, so some people, they also doing subcellular fractionation as lysis as a protein extraction, right? So they normally use a kit. For example, here I show you the subcellular proteome extraction kit called SPEC. So you can grow the cell and then use the first lysis condition to extract the cytosolic fraction, and then the next one collect the cytosolic and do the next step to collect membrane fraction, and then nuclear fraction and cytoskeleton fraction. And then you can analyze by IFA, immunofluorescence assay, or by Western blotting using a positive control for each fraction, right? So this is standard procedure for fractionation. But why do we need fractionation? Why do we need to do fractionation? And can I get all you will go back to the poll and answer the questions? You use fractionation to examine protein localizations, or examine protein trafficking, or to increase detectability of the target protein. Just pick one. What's relevant to your experience? I give you a few seconds. Okay, so most of you just choose to examine protein localization. Yes, that's the most common application. And most of you also choose the increased detectability of the target protein. That's really good. That's what I want to know. Okay, going back to my presentations. So basically, what I want to emphasize here is that there's no right and wrong answer. It's just I just want to emphasize that the fractionation that to increase the detectability of the target protein. Why that? Because if we look at the number of proteins, right, in the sample, for example, here bacteria, there's about 1,000 proteins. Mammalian cell, culture cell, about 10,000 protein. Tissue, you have more than culture cells. In plasma sample, there are millions of protein, right? However, with the analytical technique, you can detect up to 1,000 protein using mass spec, for example, or with the 2D gel electrophoresis, you can detect maximum 2,000 proteins, right? And they only detect the highest abundance protein, right? But in your sample, they are also very weak, very low abundance protein as well. So basically, the highly abundant protein just masks the low abundant protein, and you can't detect it if you analyze the whole cells, like I said. So that's the reason why we want to do the fractionation, just to increase the detectability of target protein. Okay, this leads me to the next section. That you need to do the purification, depletion, enrichment for the target protein. And in this section, I will touch on the resins, on the bit, on the depletion, the analysis, ultrafiltration. Okay, so let's talk about the affinity purification. So basically, you start with the cell lysate, and then you do the by wash dilute procedure, right? Using a ligand, using a support resin, for example, agarose resin or magnetic bit that's conjugated with a ligand, for example, here with the nickel ion, right? You can purify the His-tag protein. And then some people, after purification, they want to cleave the tag using the enzyme. That's how you purify the protein. Okay, so how do you choose whether you use the magnetic bit or agarose bit? It depends on the yield that you want to produce. With the screening purpose, microgram protein, you probably want to use magnetic bit. With the batch, milligram, you can use agarose. With the process large scale purification, you want to use the agarose or fast flow or super flow, which are also the agarose resin. And depending on the applications, for the IP Western blotting, you just want to use magnetic bit or agarose. But for the protein structure crystallization, you have to use the agarose. And depending on the analysis, FPLC, you always need to use agarose. But if you want to use spin column automation plate, magnetic bit can be used well. The key difference between the agarose and magnetic bits are agarose is much cheaper. They are more flexible. You can pack into the column, and this can be scalable, and you can use for HPLC. But with a magnetic bit, it's easier to use, and normally for a smaller sample, a low throughput. And there's one key difference here I want to show you. Magnetic bit can be visualized, right? You can see clearly because the color of the bit, but with agarose, really hard to see the bit. Therefore, when you handle the sample, you want to use the unbound material, it's really hard, it's quite hard to remove all the unbound protein from the agarose bit. Therefore, when you run the analysis on Western blotting, for example, you normally see some kind of high background with the agarose methods. But with magnetic bit, you can have a cleaner background, higher yield, high purity, and low non-specific binding. That's the key difference here I want to show you. And let's touch on the resin, affinity resins. There's different ligand chemistry depending on the application. If you want to do IP, antibody purification, you want to use protein AG, if you want to do the recombinant protein purification, there's some common tag you can use, or biotinylated molecular purification, you want to use streptavidin or ividine as a ligand. And if you want to label the protein, you can use the NHS chemistry or cumbersome chemistry. And there's some technology available for each ligand. But let's take a closer look on the recombinant purification. What are the difference between the common tag here? With the GST, MPP, they are quite large, and strep tag, his tag, flag, they are very small, right? With the large tag, they might interfere with the protein conformation, protein functions. And they have different media capacity. If you want to do large-scale purification, you want to use his tag, for example, or GST or MPP. And the purity also are different. With the flag tag, they are epitope tag. You need to use the anti-flag antibody resin. So therefore, it gives you very high purity. It's very specific. It gives you very high purities. And if you want to increase the solubility of a target protein, you might want to use your MPP. So depending on your purpose, your application, you want to choose a proper tag for your protein. And if you want to get more detail, just jump on the sigma.com/RPE website. There's a lot of useful information about how you can choose a proper tag for purification. And next one is the depletion. This is more specific to human plasma or serum sample. In the plasma sample, there's a lot of highly abundant protein like hemoglobin, albumin, or IgGs. But there's some cytokine present at a very low abundance protein, and they are mostly the very important biomarker, right? So we need to remove the highly abundant protein because they just mask the low abundant protein. And this graph shows you that the highly abundant protein just make up mostly about 95% of total protein, and the low abundant protein only make up about 1 or 5 to 10%. Okay, so how we can do the depletion? So basically, you normally use a column, affinity column with the resin, with the ligand. It can be the anti-albumin, anti-IgG, or anti the most 10 or 14 highly abundant protein in serum. So with the blood sample here, you have a mixture of protein, right? And if you don't do depletion, you run the mass spec, you can see that this is the target peak, and there's also a lot of other peak that mask the target peak. You see before depletion. When you do depletion, you run the sample through the column, so they just capture all the highly abundant protein, and then you collect the flow through, run on the mass spec, you can see that is the target peak only come up. Read the detectability of the target protein. Okay, so let's move on to the next, the last session, protein enrichment and buffer exchange. How we can do it. Okay, with this, I want to again go back to the poll and answer the last poll. What method do you use for buffer exchange? Analyses, protein precipitation, ultrafiltration. Just pick one that's relevant to your. A few seconds. Okay, so look like most of you chose the analysis. Some chose ultrafiltration, some do protein precipitation. That's really good. Thank you for your response. Okay, what are the difference between these methods? With dialysis, right? You just have the sample in the tubing and then incubate with the new buffer overnight or over the weekend. They will be changing the buffer gradually, right? It's a very slow process. Chemical precipitation, you can do the sorting out or using it's precipitated protein, but this might denature the target protein. This might kill the protein functions. And the freeze drying, same thing. You need to add a lot of salt to precipitate protein and do the speed effect, for example, just to lyophilize the target protein. This way, you also might denature the protein as well. Okay, let's talk about the ultrafiltration. What was the ultrafiltration? So in the lab, you might use microfiltration using the sarin filter with 0.2, 0.45 micron filter, just to remove bacteria, for example, or just to do the clarification. They call microfiltration. Ultrafiltration this has much smaller pore size with kilodalton pore size that you can remove protein, nucleic acids or viruses, very small particle. And the separation just based on size, based on the size of the molecule. There are different size molecular weight cutoff available from 3 to 100 kilodalton, and they can based on centrifuge or vacuum or pressure, just to push the sample through the filter, and the molecule that's larger than the pore size will be retained, right? Retained on the filter. Okay, so in the market there are two different membrane material that can be used for ultrafiltration. They are regenerated cellulose membrane, which are RC membrane, and they are PES, right? RC and PES. What are the difference between them? With the RC membrane, they has much more tighter pore structure, and therefore you normally with the ultrafiltration membrane, it's not an absolute cutoff; it's a nominal, and therefore you normally apply the window cutoff. For the RC membrane, there's the rule of 2. So you need to choose the molecular weight cutoff that's 2 times smaller than the molecular weight of the target protein, right? But with the PES, it has more open pore structure. Therefore, you normally apply at least the rule of 3. The RC membrane has the lowest protein binding, so it's reduce the non-specific binding. But the PES has a much higher non-specific binding. And RC membrane has a higher retention rate, so you can increase the yield. You don't lose the sample. But with the PES, because of the more pore structure, it can run faster. Therefore, it can lose sample. And yes, in general, the RC membrane is a more precise membrane with a higher recovery and lower non-specific binding. So I will show you the quick video when we do an experiment comparing these two membranes. We use 10 kilodalton cutoff for both membrane, RC and PES. We run a sample through the filter. We apply the pressure. And the protein sample was the red color. It's the cytochrome, about 12 kilodalton. We apply the pressure. You can see with the PES membrane, they just leak through the filter. with the RC membrane with a tighter pore structure, the protein retained within the filter. Okay, next one, I want to show you different formats of ultrafiltration. There are inverted spin filter, there are direct spin with a little bit a larger volume with the pressurized devices, they call stir cells. You can use the separate filter, right? This filter, and this is the only consumable. They can be, the system can be reusable. And the benefit of this system is that the steering, right? Steering, you can mix the sample during the ultrafiltration. Okay, let us on the purify and 2 mil ultrafiltration filter. They call imicon and microcon. The key design is that they have the vertical membrane design. So there's the desktop volume at the bottom. So you never get your sample dry. But with the microcon, it comes with the horizontal membrane design. So depending on your application, when you want to do the proteomic tryptic digestion, for example, you want to remove all the liquid before digestion, you want to choose microcon. But with imicon, you want to use for buffer change or just remove the contaminant. And with the bigger volume for they call 4 or 15 mil, you can load the sample, do the spin, remove the old buffer, you load the new buffer, you do another spin, do the buffer change, and another spin to concentrate the sample, and then you can collect the sample using the pipette. And the last one is I want to show you the Amicon Pro device that comes with, it's just the same as the ultrafiltration filter, but it comes with an exchange devices. On the top here, with the small fridge, it can retain the agarose bit, right? Agarose bit can be retained on the device. So with this device, you can do the affinity purification as well. So you can do buy, wash, dilute, and buffer change in one device with just a few spins. Okay, let's summarize about the protein purification part. I have touched on the magnetic bit, agarose bit, the difference between them, the tag resin, depletion for biomarker discovery, and enrichment using the ultrafiltration filter, the difference between the RC and PES, and the different formats of ultrafiltration devices. Okay, well, with that, I want you to take away 3 points here, just to make sure you maintain the sample integrity and compatibility, right? You want your sample to be compatible with the downstream analysis. The next one is it's not one size fits all. You need to optimize the lysis buffer for your target protein. And lastly, just get it right from the start. We need to get it right. If you can get a ton of protein extracted from sample, then it makes it much easier for downstream analysis. And lastly, just always make your protein happy. Be gentle with the protein and always keep them cold. Okay, with that, there's some resources you can access to. There's the website on the Sigma website about protein research. There are a lot of useful information about the topic I touched on today. And I just stop here and thank you very much for your engagement, for your attention. And if you have any question, please put it into the Q&A panel. Thank you again. And I just hand it over to Yi Cheng for running the Q&A sessions.
Thanks, Peter. Well done. Great presentation. So for the Q&A, I've answered most of them, but there's 3 questions outstanding. So I'll go through that with you. The first question is, what extraction solvent and workflow is best for global proteomics?
Okay, that's really good questions. Extraction solvent. and the procedure for global proteomics. Actually, I don't have the clear answer. There's no best solvent that can be compatible with the mass spec, except for the acetonitrile or methanol. And probably you don't worry much about the compatibility. You need to extract your target protein out of the sample first. Just use whatever solvent, detergents, and then you just clean up your sample. Just remove using buffer exchange. Just remove detergent before the mass spec. But with the global proteomic, I would recommend you to do the fractionation, right? As I show you, that you need to fractionate the sample just to divide your sample. Whether you want to use the chromatography for fractionation or use the method for example, extracting the membrane, nuclear, or cytoplasm fraction. That's where you can increase the detectability, right? And then you can combine the result from the proteomic on each fraction. That's where you can increase the identification. Very good question. Thank you.
Thanks, Peter. The next question is, does the salt on lysis buffers affect the protein purification step later on?
Yes, absolutely. It will affect the protein purification and also mass spec. So therefore, a lot of people, they need to do the buffer exchange to remove the salt before purification. Just do the ultrafiltration or the analysis, and you can remove the salt.
Right. Okay, last question. How do I check the efficiency of protein extraction? Do you have any advice?
Yes, the efficiency of protein extraction is largely depend on the target protein, right? There's no magic lysis condition, magic lysis buffer that can extract every protein, right? You need to optimize your lysis condition for the target protein. And the way I show you that you can use Western blotting you do the lysis condition, optimize the detergents. Each detergent start with 1%, and then run Western blotting. Just pick the best one, the best detergent, and then you can do the titration on that detergent. So that's why you can optimize the lysis buffer for the protein solubility.
Great. Thanks, Peter. I think that we'll end the Q&A now, and I'll hand over to John.
Thank you, Richie.
Thank you, E. Thank you, Peter. Okay, thank you everyone for your attendance today. We hope we've answered all your questions in the Q&A. If you do have any outstanding questions, you had Peter's email address on the screen before, so please reach out to Peter. Or also, you all have local salespeople from Merck, so please reach out to them as well if you have some questions or need some assistance in that space. Okay, so that's it for us today. Just to remind you, in a fortnight's time, we'll have our final leg of the webinar series, that being 3D cell culture, the new Lego. So we'll be hearing from another one of our field application specialists there, Dr. Peter Tran, in 2 weeks' time. So lastly, on your way out, we'd love to hear your feedback. So as you do exit, please, if you can, fulfill our survey, as your feedback exactly makes us give you bigger and better webinar series in the future. So thanks again for your time, and have a lovely day. Bye.